story that we have about this chyme that is moving through the stomach, through that distal portion of the stomach, and into the duodenum, the proximal portion of the small intestine. As soon as it enters into this proximal portion of the duodenum, this small intestine, is where it ran into all of those secretions coming from the liver and coming from the pancreas and the gallbladder, and so that's why we talked about it then. But now that it's kind of mixing together with all this pancreatic juice, the chyme is mixing together then with our secretions from the liver, now we're going to start to see the functionality of the small intestine itself come into play. And so the small intestine is named for its diameter, not its length. It turns out that the small intestine is actually the longer of the two versions of intestine. So it's about 3 meters long in a living person, only about 6.5 meters long in a cadaver. Why do we see that it's longer in someone who's dead than someone who's alive? So does it keep growing? Like you die and all of a sudden your intestine gets like starts to grow? No. That's a good guess. So it's not that there's something always in the intestines. Think about the wall of the intestines. So there's smooth muscle in the walls of the intestines. And so what do we have always with muscle? There's some level of muscle contraction, which we call muscle tone. Exactly, muscle tone. So there's always at least a little bit of kind of semi-contraction that's happening that's going to be contracting the wall of the intestine. And that's keeping it a little bit shorter in someone who's alive. Once you're dead, you don't have muscle tone anymore. So all of that muscle relaxes, meaning that you're now going to actually have a longer intestine than you when you were alive. It's about 2.5 centimeters in diameter. So again, about the same diameter as the trachea. So we said that the trachea was about 2.5 centimeters in diameter. The spinal cord is about 2.5 centimeters in diameter. And now the diameter of the small intestine is about 2.5 centimeters. It seems to be a recurring diameter. But it does extend from the pyloric sphincter. Where do we find the pyloric sphincter? Sorry? The distal end of the stomach, so it's kind of the boundary between the stomach and the small intestine. And then it extends all the way down to the ileocecal sphincter, also called the ileocecal valve. So you'll see that name being used interchangeably, a valve or a sphincter. Why do we call it the ileocecal valve? So it's going to be the boundary between the ileum, which is going to be the distal end of the small intestine, and the cecum, which is the proximal portion of the large intestine. So the name is actually telling you the two parts of the intestine that are going to be side by side and linked by that valve. Okay, so we'll talk more about the junction point between the small and large intestine just tomorrow. But for now, what we're going to focus on is this small intestine, and the big idea with the small intestine. See what I did there? The big idea with the small. Okay. So the big idea with the small intestine is that it's all about surface area. Surface area. Surface area, surface area, surface area. The more surface area you have, the more opportunity there's going to be to have both chemical digestion and absorption. And a lot of times people focus on this idea of absorption. You need all the surface area for absorption, and that's true. But we also need to have lots of surface area for digestion to happen. Because we know that we have to have digestion, the chemical digestion taking place in order for absorption to be able to work. So when we take a look then at the three parts of the small intestine, we find that this proximal portion, the duodenum, is only about 25 centimeters long. So again, not a very large component or long component of the small intestine, but arguably the most important. And the reason that I would argue it's probably the most important of the three is that most of the chemical digestion that's going to be happening is going to be happening most intensely here in the duodenum. This is where you're having all of the secretion that's taking place. You've got all that pancreatic juice that's coming in with all of those digestive enzymes. You've got all that bile that's being secreted here into the duodenum. As we'll see in a few minutes, you've got the enzymes coming from the wall of the intestine that's going to be secreted here in higher concentrations. So lots of opportunity for chemical digestion to happen. And then the rest of the small intestine, the jejunum and the ileum, are going to be very, very important. Yes, for digestion, but more so for absorption. So now we're really absorbing all of those end products of digestion. Lots and lots of opportunity to be able to get those nutrients and resources into the bloodstream or into the lymphatic system. All right, the longest portion is actually the distal portion, that ileum, about 2 meters long, and then the jejunum in between, of about 1 meter, and then you've got our full length. All right, so let's take a look then at this idea of surface area. Surface area, surface area, surface area. What's the most important component of the small intestine? Surface area. It's all about surface area. So, if it's all about surface area, that means that you never want to have a flat surface. Because a flat surface does not give you a whole lot of surface area. If you have a flat surface, you only have that amount of space in order for absorption to take place. So if, for example, I was going to say, I've got my small intestine inside my abdominal cavity here, and it's all just one big flat surface, then I've got this amount of surface area for absorption to happen. Okay, fine. So I've got an 8.5 by 11 piece of paper's worth of absorption space. What could I do to increase the amount of surface area, but not have an abdominal cavity that is gigantic? Folds! Oh my gosh, it's like you looked at the screen and saw that there's a bunch of folds here. Absolutely right. So it turns out that the inner lining of much of the small intestine is not a flat surface, it's folded. And this again is one of the reasons why we see that in the cadaver there's a much longer length, because all of these folds are going to be held in place with this muscle tone. And so when you lose that muscle tone, then all of this surface area kind of always flattens out, and then your intestines like triple, well, double in size. Okay, so these are called the circular folds, exactly right. So those circular folds then mean that we're going to be able to have a much larger surface area. So now, again, if you like to have the visual, here's my nice little normal amount of surface area, 8.5 and by 11 piece of paper in my abdominal cavity. But now what I can do if I fold the surface, I can take up the same amount of space, right, and yet have, gosh, look at that, like twice as much surface area as what we had before. It's not taking up like more volume, right, but it's giving me a lot more surface area for that absorption to happen. Okay, so we've got these permanent folds on that inner surface of the kind of large components of the small intestine, the duodenum, the jejunum, to increase that surface area as much as we can, so we have lots of space for absorption to occur. Now, if you look at the surface of those circular folds, why have a flat surface when you can have something that is going to give you an increased surface area? So even the folds themselves are not going to be flat. So yes, you've got these nice little folds on the inner surface of your intestines, but why have a flat surface on each of those folds when instead you could have these little finger-like projections, these three-dimensional finger-like projections that are sticking up off of the surface of each of these folds. And it turns out that these finger-like projections are called villi. And so these villi, these three-dimensional structures, at the center of them, we're going to find capillaries. So again, you can see one villus right there. There's a capillary network, as well as our lymphatic capillary that we find at the center. It's our green line there in the middle. And so this is where all of our nutrients that we're going to absorb are going to end up. So you can imagine that if you've got a villus, this finger-like projection that's sticking off the surface of the intestines, you can now have absorption that happens all the way around the entire circumference of this finger-like projection of this villus and all the way along the entire length. So that's going to increase that surface area quite a bit than just having a simply flat surface. So when we find these villi, we see that it's essentially just a layer of mucous membrane, so these absorptive cells that are going to be forming the outer lining, epithelial tissue, And then a little bit of connective tissue. That's this lamina propria, this connective tissue at the center, which is going to contain our blood vessels. We know epithelial tissue does not have blood vessels associated with it. It's connective tissue that has our blood vessels. And so that's what this lamina propria is. It's the center kind of core portion of this villus that has our blood capillaries as well as our lymphatic capillaries. In between each of these finger-like processes sticking up from the surface of the wall of our intestines, we have our intestinal glands. So these intestinal glands are going to be responsible for secreting intestinal juice. And as we'll see, intestinal juice, yes, mostly water, but we're going to have some enzymes that are going to be released from here as well to help in this chemical digestion. When we take a look then at the villus and kind of zoom in a little bit more closely, we see that they are lined with this epithelial tissue. So you've got these epithelial cells that are going to be forming the kind of the surface of those villi. And those are going to be our absorptive cells. Those are the cells that are going to be able to take the nutrients from the lumen of the intestine and then move them through into the center where those blood supplies are going to be found, those capillaries are found, and then we're able to have absorption happen. But on the surface of those cells, what do you think I'm going to say? Why have a flat surface when you can have a folded surface? So even the surface of the cells themselves are not going to be flat. They are going to have these kind of undulations or kind of folds in the surface membrane. Okay? We're not talking here about cilia. This is non-ciliated, simple columnar epithelial tissue. So we don't see those hair-like processes that are kind of weaving back and forth and rhythmic motion. That is not what we're talking about here. Instead, we're talking about microvilli. All microvilli are folds on the surface of the cell membrane. Rather than your cell membrane just being super flat, like you normally draw a cell, we would draw the cell membrane as kind of just having these big kind of dips and grooves, and then it forms these little kind of processes, these little microvilli that are sticking out on the surface. Okay, but they're not a separate structure. It's not like a cilia, which is a separate kind of hair-like structure. So these microvilli are very densely packed together. About 200 million of them per cubic millimeter. Per square millimeter. So again, if you're looking at 1 millimeter by 1 millimeter, it's about 200 million of these tiny little microvilli. So again, lots and lots of surface area. Each microvillus, then, you can have absorption that's happening all the way around each one of those little processes. Lots and lots of surface area. What did I say the whole point of small intestine is? Surface area. It's all about the surface area. So this inner lining of the intestine is not flat, right? Those folds themselves are not flat. We have the villi. The cells on the surface of those villi are not flat. So it's all about increasing the amount of surface area for absorption. These microvilli, because they're having these kind of folds in that surface membrane, look kind of like a brush. So we're going to see an image of it in just a second. But it really does look like the little tines on a brush. And so this area, this kind of surface area of that cell, is what we call the brush border, because it looks kind of like a brush and it's right along the border of our intestinal wall. And so that's where we're going to find a lot of these digestive enzymes, and where absorption is going to be able to take place is right through those absorptive cells and then into the capillaries that we find at the center of the villus. We take a big step back and say, okay, what does this actually look like inside the intestines? You can see, first of all, this is part of our duodenum here, and you can see all these big folds on the inner surface. What are all these inner folds called? Those are the circular folds. That's these, right? The circular folds, so it's not flat on the inner surface. Then, if we zoom in onto one of these individual circular folds, and actually you can see that, I like this image because you can actually see that the surface of that fold itself is not flat. Right? Instead, what is it made up of? What are these called? Those are the villi. Those are those finger-like projections. And at the center of each of those villi is where we have our capillaries. Okay? So then here you can see those villi all kind of standing upright. Right? You can imagine absorption, digestion and absorption is happening all the way around the circumference of each one of them, and then all the way along the entire length. So lots and lots and lots of surface area to allow for this absorption to happen. Then, and again, you can recall that at the center of each of these villi, that's where you're going to have your blood capillaries and that lymphatic capillary there as well, on each one of them. So that's where all of that absorption is taking place. And then if you zoom in even closer, you can look at the individual cells that make up the wall of this villus, all of these villi. And what you find then is that we have our absorptive cells. And that's what we see here. This is actually two cells side by side. You can see their cell membrane is all nice and tightly anchored together. This is the apical surface. This is where the chyme is going to be moving past. And you can see all of these little structures here. What are those? Those are the microvilli. Remember, they're not cilia, they're not little hair-like processes, they're just inundations, or I should say, not inundations, that means they're flooding, indentations of the cell membrane, and then it gives you this brush border-like appearance. So it looks very much like a comb or a brush, and it looks a little bit kind of fuzzy or furry on the surface. That's the brush border that we're talking about. All right. That's the absorptive cell. That's the cell that's going to allow for that digestion at the surface and then absorption of the nutrients through that cell and into the core where that blood supply, where that capillary network is going to be found. But we have a couple of other cells that also make up the lining of the small intestine. We have goblet cells. We've seen these before. Goblet cells. This is a mucous membrane, so we're going to see that there is mucus that's going to be produced and secreted into the lumen of the small intestine. We see that there's going to be our enteroendocrine cells. Hey, what do these enteroendocrine cells produce? Hormones, because that's what it says there, but which hormones? CCK and secrete. Hey, are two hormones that you don't know anything about because you haven't done your assigned reading. So when you go back to do your assigned reading and you figure out, well, where are these hormones produced, this CCK and this secretin that we keep talking about, these are the cells that are producing those hormones. And then we remember that those hormones have three different functions, one on the stomach, one on the pancreas, and one on the liver. And then finally we have these Paneth cells, which are going to be kind of modified versions of white blood cells. They act as phagocytes, kind of clean up any debris that might end up in those intestinal glands. They also secrete lysozyme, which is a bactericidal enzyme that helps to break down any bacteria that might be there, kind of help to prevent any major infections. All right. So those are the cells then that form the wall of the small intestine. This then brings us to the two types of digestion that's going to happen in the small intestine. The first, of course, being our mechanical digestion. Mechanical digestion in the small intestine comes in two varieties. The first is the migrating motility complex. This is the one that we often think about when we think about movement of chyme through the intestines. This is going to be this relatively slow kind of contraction, peristaltic contractions, nice kind of really smooth movement of this wave of contraction along the length of the intestine. Once we start one of those waves at the distal end of the stomach and it passes then into the duodenum, it takes anywhere between 1.5 and 2 hours to be able to travel along the entire length of the small intestine. So again, a very slow moving kind of wave of contraction that's going to be moving along. Overall, the chyme takes anywhere between 3 and 5 hours to work its way from the stomach along the length to the large intestine. So if here's some chyme, you've got this nice wave of peristalsis, and so you get pushed along a little bit, and then the wave carries on. And then another wave comes along, and you get pushed along a little bit further. So you, as the chyme, are moving a little bit more slowly than the wave is, but you're kind of constantly being pushed further and further along that length. So that's great to get stuff moving along, but you also want to have the capacity to have some mixing and churning happening a little bit more locally. You want to have some mixing and churning with the intestinal juice secretions to be able to make sure that all of the chyme is at some point coming into contact with that brush border. Because if it doesn't come into contact with the brush border, well then it's going to be a lot harder to absorb any of those nutrients. And so we have segmentation. And so segmentation is where we have this localized kind of mixing back and forth in kind of a smaller region to make sure that we have that churning and mixing that happens to bring all of that chyme into contact with the wall of the intestine. intestine. This is a little bit faster, so now you're getting kind of 8 to 12 kind of sloshes a minute in each of those areas, and it helps to churn things along. As you're churning, you're kind of slowly getting pushed along, so you've got kind of two motions that are happening at the same time as that kind is moving along the line. Exactly, so the brush border, that was microvilli, where you have all of those digestive enzymes and where that absorption is going to happen. That happens along the wall of the intestine, and so you want to make sure that all of the chyme at some point comes into contact with that brush border so that we can have that digestion and absorption happen very efficiently. Okay. So we talked about this idea of peristalsis, and we mentioned peristalsis when we were talking about the esophagus, and we mentioned peristalsis when we were looking at the stomach and kind of that contracting, and now we've mentioned peristalsis once again, but really what does peristalsis look like, and what is peristalsis? Well, as I mentioned a minute ago, it's this really kind of smooth contractions of the walls of the intestines and the stomach. And so I always remember peristalsis from my high school biology teacher, who was a fantastic teacher. He used to bring in all kinds of, he knew a guy who worked at an abattoir. You know what an abattoir is? Where large animals go to die.
Yeah.
So like horses and whatnot. And so he used to bring in horse hearts into our biology class. You guys get to look at real human hearts, all right? We didn't have that. We had horse hearts, horse eyeballs, and horse lungs. You guys get the real thing upstairs. But all we had were horse parts because he knew a guy. So he went to the abattoir. I'm not sure how legal that was, but whatever. So he went to the abattoir one day and asked his buddy, well, what's in the big green bin over there? He said, well, go have a look. So he climbed up the little ladder and then threw open the lid of this big green bin, and inside, he said, was this big pile of pulsing intestines. So just a gigantic bin full of these horse intestines that were all kind of doing their last little bit of peristalsis. So the moral of that story is never look in the green bin. And peristalsis really is kind of this rhythmic, nice and smooth. It's not jerky contractions like you see with skeletal muscle. It's very smooth and kind of controlled contractions of the wall of the GI tract. So while I don't know a guy, but what I do have is a video of some peristalsis. So this is actually in the stomach, the distal end of the stomach, and you're going to see that this wave of contraction is happening, coming kind of from behind us, and then squeezing through. But you see how smoothly this kind of contraction rolls along, right? And then you can imagine that if you're a chyme in there, you're going to kind of get pushed along, and then here comes the next wave of contraction. But it's nice and smooth and slow, and there comes the next wave in behind it. And so this is what we're talking about when we're looking at peristalsis, right? So again, nice rhythmic contractions. Poor horses in their green bin, but that's peristalsis. Oh, that's such a cool video. Okay, now, when we look at the chemical digestion, so that's mechanical digestion. When we look at chemical digestion that's happening in the small intestine, we have to recognize that we've already started a little bit of chemical digestion taking place. A little bit started in the mouth, a little bit started in the stomach, but really it's mostly going to be happening now in the small intestine. Now that we've got all of these enzymes, especially for the pancreas, along with the bile, now we're really going to be able to have that chemical digestion get up and running pretty efficiently. So when we take a look then at the three different categories of components, we find that we'll start with our carbohydrates. So in the mouth, as we said, salivary amylase. How many of you did that nifty home experiment to prove to yourself that you have salivary amylase with a cracker? No one ever does that in my nifty home experiment. I don't know why. But anyway, okay, so you've got a little bit of salivary amylase. Again, not super efficient. Just don't let it sit in the mouth long enough. And so really it's going to be in the duodenum. So we've got our pancreatic amylase that we talked about before. It's going to break down some of those starches, and then we've got some of those intestinal brush border enzymes produced by the intestinal cells themselves that help to get us down to the level, the smallest level, those monosaccharides, things like glucose, fructose, that are going to actually be able to be absorbed. What about our protein digestion? Well, as we said, protein digestion, we're happening in the stomach, and so protein digestion that is going to occur is going to be a result of that pepsin that converts those proteins into those peptides. Okay? But then again, to get to kind of the final end product, we're going to use those pancreatic proteases that get us down to the amino acids, and then once again, those intestinal product order enzymes that get us down to the amino acids. Because it's really the amino acids that are going to be the most easily absorbed through the wall. All right, and finally we have our lipid digestion. So lipid digestion then, as we said, begins in the mouth and the stomach, but we have those two types, which are pretty weak, right? We've got the lingual lipase, the gastric lipase, not super efficient. Most of this digestion for fats is going to happen in the small intestine because of that bile, first of all, being able to break down those big globules into small globules, and then of course the pancreatic lipase that is going to further break it down into those subunits that we can absorb, the fatty acids, and those globules. Great. So I think what we'll do is stop there today. I can feel the anxiety starting to rise.